follow up on Omega Omega Bio-Nexus. Please take care of your phone. Put your phone on silence. So please, let's go to Omega Bio-Nexus. If you are at Omega Bio-Nexus, can I see you? Can I see your hands? Right there. Okay. Who again? Who again? Where is Richard? Are you there? Timileyin. Who is Timileyin? What's your name? Lamidi. Belumi. Okay. I shall know is okay. Right there. I only have 3 people watching, no, including me. Nobody's there. So before we will go on, before we go further, to don't forget our major objective is to make sure that we install Panaro. Now, by now, you should know. Let us go to my workflow again. Let me display the workflow, and so that you'll be able to know at which point are we. For some of us that might not be there, don't forget that this is a work of somebody who is planning to move from wet laboratory to dry laboratory. That is somebody that started with field work, which has to do with so when we talk about moving from wet to dry lab, it will include the field work, it will include the wet lab, which is the laboratory, and it's going to include the dry lab, which is going to be on computer. The field work will be on the field. You cannot do it alone. You need some major stakeholders. You need to make consultation. You need to prepare very well, because what you are going to do on the field or in the field, you might not have opportunity of doing it again. Any mistake you do on the field, it might actually cost you that you need to go back. So that is why we said that this level, you start with number 1, which has to do with general sampling, which is pre-sampling, which involve identification of major stakeholders, advocacy, consultation. Then you prepare for your instrument, which could be in form of a questionnaire. This person prepare for questionnaire, recruitment, got ethical approval, and so on. Then at a point, he was able to map out where it's going to be his target site, which was done in at least 4 different states. That is in TB centers in those states, Oyo, Ondo, Ekiti, and Kwara. So after the collection of that sample, it was transported for processing in the laboratory. That is, we are already moving from the field, and now we are moving to the laboratory. So part of the processing that was performed or were performed in the laboratory include the contamination of that sample, which has its own function or rationale behind it. Another one is the use of GeneXpert. That's going to divide your work. At that level, you'll be able to identify the TB into drug resistance and sensitive drug resistance. So at that level, then you perform the smear microscopy, which has to do with the use of acid-fast stain, AFB stain, so that you'll be able to monitor the way Mycobacterium tuberculosis is looking like under the microscope. Don't forget that under the microscope is going to be, because it belongs to the family Bacilli, so you will see how it is going to be under that. So, and it is going to stain positive for AFB stain. So, and don't forget that one of the differences between gram-negative, gram-stain bacteria, that of acid-fast stain, is that in the cell wall of acid-fast, it has additional structures, which include the mycolic acid, muramic acid, fatty acid, and a lot like that. So that made it very, very difficult for any ordinary stain to be able to stain it. Then after that, you now go to isolation and culturing. And this one, don't forget that Mycobacterium tuberculosis is highly infective because it is an airborne infection. And apart from that, it is at the BCL3, that is biosafety level biosafety level will require you to be very, very careful in handling it. So that's why you don't just have anybody dealing with Mycobacterium tuberculosis just like any other one, because it is highly effective. So even in that, after that one, you now enter into the molecular analysis, which has to do with DNA extraction, PCR, library preparation, and I told you what a library is. Then that one also led us to whole genome sequencing. So, and I said, in this case study we are considering whole genome sequencing from Illumina technique. I gave you an example, I mean, a kind of homework that you should be able to identify different types of sequencing techniques and what are the advantages and disadvantages of using each one of them. Then what are the rationale for that? But don't forget that we are picking whole genome sequencing because we want to know, we want to study the genome, the totality of the genes on Mycobacterium tuberculosis. So after that, um, we now enter into the dry lab, which has to do with genomic analysis. Um, we don't forget you register at Galaxy on that. Then I told you that Galaxy is an incorporated, it's not a standalone database. That is, when you are still under Galaxy, you will still have the opportunity of making use of another tools. And I told you what we mean by tools in the physical. That is, if you don't have the right tool, you might not be able to do you might not be able to perform the task. So tools are actually used to execute specified tasks in bioinformatics. So, and I told you that if you want to mend your clothes, the best tool you should use is just needle, not knife. So the same thing with bioinformatics. If you are to perform a certain task that will require you to have to make use of one tool, and you don't have that tool, you might not be able to proceed to the next level. So one of the tools that you need to get acquainted with is um, Galaxy. And I told you that irrespective of wherever your sequencing is coming from, there are going to be some errors that will come from the sequencing machine itself. So, and there are some errors that are going to be like adapters. They will adapt themselves. So all these errors are part of your raw data, the raw data that you are going to have from your sequencing machine. So, and one of the first thing that you need to know is to determine the quality control. What do we call the quality control? Quality control is to determine how reliable your own data is. And it is very, very important in science that you do the quality control. So that quality control, one of what we do is that you just go to the Galaxy and you just go and type uh, faster QC, FastQC. That is the tool you will use to clean your data. Don't forget that I told you that your raw data is going to be in form of FastQ. Okay, so you run FastQC on your sequences. Then after that, you'll be able to trim your data and clean the data using fastp or Trimomatic or cut adapt. Now, because we are going to use de novo comparative genomics for this system, the most advanced and the most recent, the most modern out of these three is just fastp. So if you cannot use fastp, you can still make use of Trimomatic. So that Trimomatic is going to do the same thing. Same thing. Trimomatic is just like a tool, like scissors. It's going to cut away all the adapters. I told you that the adapters will be like some of these iron filings that if you are looking for you are looking for your needle in the hay. So what you do is that your magnet will not only pick only the needle, it's also going to pick non-needle but which are also made up of iron. So this adapter can be likened to something like that. So after that, after the trimming, the next thing is to assemble your gene, bring them together. Don't forget that when you have your raw data came in form of a two. It came in form of reverse and forward sequences. So by the time you have trimmed your data, that means you have cut at each end of the strand, or from the reverse as well as from the forward. It's going to trim it. So after that, after doing that, so the next thing is to join the two strands together. That is the reverse as well as the forward. When you now want to join them together, you need some of these tools. One, for genomic assembly, you need spades, or velvet, then or Mykrobe. You can also make use of a genome analyzer. Then you can also use BVBRC or Combat TB. These are very, very specialized tools that you will actually need if you are dealing with Mycobacterium tuberculosis. Then after you must have joined all your sequence together, after you must have assembled them now, the next thing is each of these genes, how will you be able to know the function of each one of them? How can you identify the function of each of them to this? So that is why you need another tool, and that is at the level of PROCAR, which I think all of you must have done that as well. Now, before we now go on, we have gotten a tool that is um, that is in charge of assembling our sequence, bringing them together. Gene annotation, we use PROCAR. And another thing that we also need to know is that of the Panaro that we talked about. This one is a pangenomic analysis. And what do we need pangenomic analysis to do? Pangenomic analysis is going to help us to identify some very key proteins or key genes. For example, somebody that is interested in designing vaccine, now you need immunogenic genes. That is, these genes and proteins that have the capability of eliciting or triggering an immune response. So if any of your protein, so you can have 5 million proteins, 5 million genes, but if you do not have the one that is going to trigger an immune response, that means you might not actually use it those sets for vaccine design. So that was why we now look at how can we how can we do pangenome analysis, and that is why we need the Panaro. And well, I'm going to take you back to Panaro now, so that you'll be able to follow. I want you to prepare your system for that task. I told you that it was not an easy task, but with ChatGPT, everything is almost very, very possible. And that's the same way we are telling you nowadays what you need to know, as a kind of a takeaway from your bioinformatics journey is that number one, your life should not remain the same. The same because number one, you don't have to struggle with so many things. So many things that in those days, before you will be able to even run all these just one installation of one package. If it were to be before, if you want to install Galaxy on our system, we have to go and buy the disk. And that disk alone, you know how much it's going to cost you. Apart from um, buying the disk, you also need to hire somebody that is going to help you with the installation. At that time, if they say one package is just 20,000, you don't have any argument about it. So that is that. Now, before we go into Panaro, let us see why do we need Panaro. So let us go back to ChatGPT and let us prompt it so that it's going to give us some insight into why. Because just like I told you that I spent a lot of time. Apart from that, it also required a lot of experience in the sense that although you might not need to be a computer person or a technical person, but if you do not know how to follow prompts, you might find it very difficult to do that. So number one, I want to prompt ChatGPT now that why, okay, or what are the rationale rationale behind uh, behind pangenome pangenomic analysis using what Panaro. Who is that? Use Panaro. Okay. So let's see what ChatGPT is going to tell us about our journey. So why do we have to go through a lot of these technicalities? So now this one is telling you, please, can you see me? Okay, this one is telling you number one, ChatGPT is telling you what is pangenomics. Pangenomics. I am trying to tell you this so that you'll be able to, by the time you are compiling your write-up, you will be able to do all this and justify. And if you want to be alone, you will not say that, me, I don't know. You just ask me to run it, and that's the same way I saw it on the internet. So you should know why you are doing what you are doing. Why are you using that tool? So when you sequence many strains of the same bacterial species, e.g., Mycobacterium tuberculosis, E. coli, Staphylococcus aureus, you will notice that they share common genes in they have common genes in common. Sorry, they share some genes in common. That is, you can find some genes that are very common in TB, the same gene in E. coli, the same in Salmonella, the same in some, because they belong to the same family or they have a related species. Then they have some genes that differ. That is, that differentiates each one of them from them. So the pangenome, what pangenome will do for you is all gene present across all strains of that species is going to get them together. All. So it will now divide this into number one, called genome. So Panaro will help you to divide them into core genome. These are genes that are found in every strain. So far they are bacteria, you found that gene in common. So it's just like the case of human being now. Now you know that, let me pick mammals as example. When you pick man, you pick apes, monkey, you pick fish, you pick reptiles and so on. There are some common genes that you will find so far that they are mammals, they belong to the same class. So all these genes will be found in them. All these are called the core genome because these genes are found in every strain. So then it is what now differentiates mammals from apes, because it is going to be too particular to that species. Another thing is that they are called essential, they have essential functions. They perform essential functions. So, and that's why they belong to the species identity. That is what they use to identify them across the species. Number two, the Panero and what is called pangenomics is going to also have another category of genome that are called the ancestry genome. These are genes that are found in some but not in all strains. For example, the genes that are responsible for drug resistance, they are not found across, they are not common. In the one the Mycobacterium tuberculosis is making use of is different from what E. coli is using. So these are called the ancestry genome. Then the one they use for adaptation, because they exist in different habitats. So, and I told you that at the beginning of this class, or at the beginning of this workshop, that you should know that when you bring a particular organism outside its natural habitat and you now introduce it into a new habitat, that means that that organism ability for that organism to survive will depend on the survival of the fittest. And I told you that in bacteria, what they will do is to change their molecular arrangement. It has molecular basis. So these are called the ancestry genome. The genes that are found that are responsible for drug resistance, those ones that are responsible for virulence. So if somebody is infected with E. coli, it's quite different. The virulence will be different from when it is infected with Mycobacterium tuberculosis. It will be different from when the person is infected with a Salmonella typhi. So all these are degree of virulence. And what do we mean by virulence? Virulence is an ability or the degree of infectivity of a certain microorganism. Then the third one is that some of them are called the unique genes that are found only in one strain. Only in one strain. They are not found in another species, but they are found only in one strain. So genes found in one single strain are the one that will be responsible for mutation. I told you one of the tools that we have been using before, we differentiate organisms based on their mutational changes. Some of them will be differentiated. There are some microorganisms that have plasmid, and there are some bacteria that do not have plasmid. In the molecular genetics class, you should know at this level that organism or bacteria that one of the differences between bacteria that has plasmid and the one that do not have plasmid is the presence of that plasmid. Plasmid is what they use for drug resistance. So when you have a bacteria that have plasmid, that means a it is very likely that that strain of microorganism or bacteria is what? Drug resistant. And the one that do not have, that means it is going to be very sensitive to this. So this the issue, in short, pangenomics will help us to understand the genetic diversity of species. So that means if we do not run the pangenomics analysis, and from running our procar, the next thing we jump to the next one without going through all these rigors. That means all this might not be so possible for us. So that is what uh, pangenomics will do for us. It is going to help us for genetic identification or understanding of genetic diversity of species. So now you already have your products of procar or bacter. You already have it now. So after having it, the next thing is to do the pangenomic analysis. So why Panaro? Why do we use Panaro? Panaro is the next generation pangenomic tools built to solve found many problems associated with the old software. There are some other software, if you say, oh, going through Panero might just be too strenuous, too hectic, and all this one. That's there are other tools that you can use. So you should know that number one, Panero has a kind of it has solved some problems that are associated with the other one. So it is widely used because uh, it produces clean, accurate, and reproducible pangenomes. So that is the main reason why we are opting for Panaro and not any other one, because it's going to give us clean data, very accurate data, and reproducible. That is, you can easily reproduce it at any point in time. Now, what are the most what are these major problems that Panero has actually helped us to um, solve? Most pangenome tools rely directly on the GFF file, which is produced by Annotation like Procar. Like Procar. Don't forget that in this work, we make use of a Procar for our gene annotation. So the product of Procar will now be the one that you are going to input directly into Panaro. So some genes are split incorrectly in Procar. So you cannot just use Procar result directly like that and you think you have already done what is actually necessary. No, because some of these genes are split incorrectly, and some genes are going to also be duplicated. There are some genes that you found them in Procar, that such gene will still be duplicated. Some genes boundaries are wrong. The annotation of some genes by Procar sometimes might not be 200% correct. So then some genomes contain bad assemblies. Don't forget that we assembled with the use of spades. So at that point in time, and don't forget that the product of spades is what we uploaded into Procar. So if there was wrong assemblage of any gene, wrong assemblage will give wrong interpretation, wrong annotation. So all these are part of some of the errors that are associated with other software or other database, especially the product of a Procar. Now, why Panaro now? Panaro fixes all these error. it will fix all this error. Number one, because it is going to use a graph cleaning. And the error in that graph, you will see the error correction that you needed to remove. That is why the product of a Panaro for you is also very, very important. So you should be expecting a graph to at the end of the day. So that graph is going to give you inflated gene counts. That is, if some genes were wrongly or inflated, they were wrongly inflated, and that in that the product of Panero, you'll be able to see it. Then broken genes, you identify it. Frame shift, you know what frame shift is now at this level, because it is a type of mutation that has to do with either deletion or insertion, which is going to lead to a shift in protein sequencing. So then it's also going to do erroneous annotation for us. It's going to tell us specifically that what PROCA, the result of PROCA, these are the percentage of the errors in that annotation. I don't know if you are following me. Okay. So all these one, you can also know, because Panaro is more accurate for bacterial species with low variations. Huh? Tools like Roary or older methods struggle with bacteria that have. Now, you need to, I told you that you don't just, as a bioinformatician or as a computational biologist, you don't need to just say that what you just need to do, just rely on it. You should be able to know. You should be able to know the biology of the organism you are dealing with. For example, you want to design drug against certain bacterial species, and you don't know their biology. You don't know their level of infectivity. You don't know the prevalence. You don't know anything about them. You might not be able to do it. It's just like somebody who is a tailor, and that person does not in any way know you, doesn't take your measurements. It's just on assumption. You just discover that maybe just through your name, just say, this is Olayiwola Adegbite. And when, because you are Olayiwola, say, don't forget, don't worry. I am an international fashion designer. I know what is going to fit that person. It might not be possible. So that's the reason why you need to know. You need to have the wet analysis about the organism you are working with. Even if you are going to do dry lab work, please, you need to know that. So, and you should know that Mycobacterium tuberculosis, they have highly conserved genomes. Highly conserved genomes. And if you want to know, what do we mean by highly conserved genomes? That is gene that they are very, very stable. They are very consistent. They cannot just undergo any mutational changes with time. So what they do is just that at any point in time, you just discover that they are very stable. They are consistent. And Mycobacterium tuberculosis is one of the most stable across all. They have highly conserved genome. Then apart from that, they have low mutation rates. Then low recombination. So that graph is going to help you to be able to solve all these problems. So, and just like I said, these are very, 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 very important. Now, Panaro is now very perfect for detecting ancestry gene variation. Is the most suitable because it is going to identify antibiotic resistance for you. If you want to talk about antibiotic resistance, you make use of it. And one thing I want you to know is that this installation of Panaro, we are going to, in the next 3 minutes, we are going to start with that ChatGPT prompt, and you will start implementing it. But you will just see that inside that Panaro, you will install more than 3 or 4 other software inside it. So which include TB profiler is there, okay, you Proca is also there, Bacta is also there. So it is part of that. So if you are working with anything like antibiotic resistance, you need Panaro. If you are working with virulence factors, you also need that. If you want to know the genes that are responsible for environmental adaptation, you also need that. Then if you are looking for plasmid and mobile elements, it is very, very useful. So it is going to tell us gene presence and gene absence. So the genes that are present, it is going to tell us, and the genes that are absent is going to tell us. So these are part of one of the major reasons why we need Panaro. Okay, why we need it like that. Then apart from that, it is also going to help us as a bioinformatician. By now, you should know how to draw a phylogenetic tree. You should know how to interpret phylogenetic tree. You should know that under normal circumstance, if you want to know how conserved or how related some organisms are, you need to know them with their phylogenetic tree. So without wasting our time, these are the reasons why Panero, irrespective of what might be the rigor, what might be the level of challenges, technical challenges that we are going to encounter, this is one of the reasons why you should go for nothing but a Panero. So I want you to, I'm going to display um the I want you to see some of the workflow that you need to know if you want to install Panaro. Okay, so and it's going to be according to what I use by ChatGPT. So please, I think you can now start from here. Okay, yeah. So the question, what I prompted ChatGPT was that I asked ChatGPT how to move from here. Where is that here? Huh? Where is that here? Is that okay? Let me see. Let me see. Uh, I could see some of you. You already have a miniconda or anaconda files. Do you have it already? Hmm? Are you sure? Are you sure? So okay, you know what? Go back. Just follow what I display for you now. Start it from the beginning. Command to install Pangenome Panero, and how and where do I get the terminal to enter the command. So it's displayed for you. So I want you to start from there. Start from there. So start from there. Make use of your. Hmm? You see what is displayed there now. Don't forget, I want you to also be jotting while you are going. You are also doing all this. Please, you have to reduce your focus on what you are to do. Just follow the instruction I display for you. Huh? So after specific time, I'm going to also move. Make sure you move along with what I am saying. You practice it, whether you are getting the same thing as you need to get. And as you are getting it, if you are not getting it, even if you are not getting it, what should you do? Just screenshot the level at which you are. Go and paste it on ChatGPT. Huh? Paste it there. So you have to open like 2 places for ChatGPT. There's one you are going to be using to be following me. Then the second one is if you encounter any problem, how do you go from there? Okay? Follow that instruction. You want to start from the scratch. Where exactly are you? Have you opened? Have you command? Okay. So what do you have? What is it? What is it downloading? You are not downloading yet. You are not yet downloading. So you have to sign in. Sign in first. It's the same thing as anaconda, misconda, miniconda. Huh? So copy that code from ChatGPT. You don't know what you will do first. Just follow it. But normally, what you are going to do first is that you are going to install Miniconda on your system. So when you install Miniconda on your system, the summary of what you will do is you will now start all those codes. It is when you have opened your conda, you just be pasting it on the terminal of Miniconda. But if you don't get to Miniconda or Anaconda, you might not be able to paste anything, even if you are seeing any code at all. So the first thing is to make sure that you know how to get to install Miniconda. And what you said you installed yesterday was not. It was not. In fact, if you have anything like that, I want you to. That's why I said you should start from the scratch. Now, so option A, you now choose. The option A is that If you want to install conda, which is recommended and is the easiest, if you already use anaconda or miniconda, that is, if you already have miniconda on your system, you just run it. So the next thing, go to miniconda, go to anaconda. You are there. And you accept now. Accept all. After you accept, scroll down. You'll see where you have 64 MB. Okay, click on downloading. Download. Go scroll down. Click on this Python 3.13. Python 3.13. Where is this? This is Anaconda. Okay. Are you be following me? Click on this Anaconda. Anywhere you have this, that's the one that is clickable. Accept. Windows 64. And one requirement. You know one PC requirement. PC means your laptop. Before you will be able to know whether you should go on, is that you must have nothing less than 3 gig. Nothing less than 3 gig space in your system. You must have nothing less than 3 gig on your system. If it doesn't have up to that, you just discover that it might not be installed. You'll be having problems. It's like when you want to fill a so make sure that is what you have. You are in another place now. This is okay. Sign for download. Why can't you change this? It's not in English now. Can't you see it? So how do you want to understand what you are saying? Language. So it's better you if you okay, it's already downloaded anyway. But if you stay on it for a long time and you start playing, get where you land. Is that immediately after you must have installed your Anaconda? Whenever you see that the Anaconda is already installed, now go to search. Where do you have search? Go to search and search for Anaconda. Search. Type Anaconda and search for it. That one will give you an impression if you already have it or not. Here, it's just you type it here. Here, type Anaconda. If it is on your system, it's going to bring it for you. Anaconda. That means it has not been downloaded. Normal. Anywhere they ask you to enter your group account, it's better you use group account. Gmail. Please be mindful of not touching. Okay. That's how it goes. So you are not following again. I won't be moving from here to here, from pillar to post. So I said you should follow my video on YouTube. Follow it. Install with pip, although not recommended. So that is when you must install your Anaconda and just open at the terminal in Anaconda. Install your Anaconda first. Then in Anaconda terminal, you should do what? So if you cannot get to Anaconda, just screenshot the message you have and go and paste it on ChatGPT. That how do you move from that place to installing Anaconda on your system? It is going to give you direction again. Has it downloaded? Yes, sir. How will you know that? Where is it? No, you come to this place. Click on this window and search. That search. This lens, okay? Type Anaconda there. Just type. Okay, click on it. So now you can now begin the journey as you have it on the YouTube. So it is that the first code that I display. Just go and copy that code and put it here. You will be following it. I don't believe in this. Don't have to do it. And the way you know, come to this place, come to this place, type Anaconda. It's not complete now. So start the journey. Next, click on next. Click I agree. And then click just wait. So next. It's not empty. Okay, now change this to so install, click on install now. I think at that point, what comes as you? So go to come to this place. This book, type Anaconda. Type it. This one will tell you whether this is still telling you. Have you searched for it? Anaconda. Anaconda. At the same level. They are at the same level. Now, the next thing is where do you get the terminal to type all those commands? All those commands, the first code, which has to do with conda install, C, conda, foxy, and so on. Okay? How will you install it? How will you install it now? Pip install Panaro. So you now follow this step. Option 1, follow the that step, or you follow window without Anaconda. Now, at this point, there are two things that you have options. The first option is that if you already have Anaconda on your system, you use the first option. That is, you press start menu, the one I just took you through now. Type Anaconda prompts, then you open it. Type the Panaro command. If it works perfectly, fine. You don't need any other one. But if you do not have, let us assume that you do not have Anaconda on your system at all before. If you have done everything and it's not already installed, you use option 2. So what is that option 2? Is that you will use that download miniconda. You will use download miniconda and are going to use Anaconda prompts. Okay. Now are you following? Follow it on that YouTube and follow the prompt I've just given you now. That if you have Anaconda on your system, you use option 1. If you don't have Anaconda on your system, you use option 2. Use option 2. The option 2 is that same command. So you can put it on ChatGPT to give you the command without what? Without Anaconda on your system. And it should give you the link to install Anaconda there. Forward it. That's before we started the computational distance. This information I just gave you. Or is this installing already? You have 2 options. They are giving you the first option that if already you have Anaconda on your system, follow option 1. If you don't have it on your system, follow option 2. Say yes. Run now. Run. For your system, this cannot be installed here. You only need the Anaconda software can only take 64 bits. and your system is 32. So that means you cannot install it on the system. You know, I told you that there are some systems that are not compatible, especially all those old systems. You can need to install your own system. I think this also recent. The system is also recent. That's this. Is it 64 bits? You have to pray. Code. You say you don't have the code. This is code now. That's the code. This code. That is command to install. Follow this. This is the first option. Your first option is that already you have an anaconda already on your system. So you are going to make use of this code. But how will you copy this code? Is that's why I said you should open another one, another ChatGPT, enter this. The prompt I gave, the prompt I gave to ChatGPT is going to give you this so that you'll be able to copy from that and install it on your. What is wrong with you? You cannot sign up since morning. They are still battling with password right now. Don't allow people that didn't even show up to be better than you. So you have to show all seriousness. Please put on those fan, please. Put on the fan. This one from here. Anyone, all of them just put them. You know, if we cannot install Panaro, we cannot move forward. And don't forget that I've already installed it on my own system, but I have to wait for you so that you do not get lost. But even if you just only 2 or 3 people that can get it to the level I have, I think others will not be able to do. That's why I said the way you are going, I'm expecting you to be jotting some things down. Jotting some things. Anyway, you see some of these things, this information again. Gmail instead of Gmail. Don't you have Gmail? So what is it? That means you don't have access to your Gmail. And under normal circumstances, it doesn't even supposed to be as if just click on sign this now. Are you sure this thing is why is it? I need to start it. Start it. Start. Good to know how to start. Why is it taking this long? Please cancel. Why is this? That's why I don't know all these things. I don't know. Start it. Charlie, why is that one time? You started again. You started all over again. Okay. What happened to it? It's what? Panaro. Let me see. Please not charge. You are not in charging it yet. You are still on Google. See. The other time when I told you that sometimes you can be on just one task for 2-3 days. Sometimes you can be on it for 5 days, 1 week. So, but the good thing about it is that if you are making progress, if you are doing the right thing, you know. And at any point in time, when you are not even, you are appearing not to even move at all, always screenshot it and put it on ChatGPT to troubleshoot for you. Let ChatGPT troubleshoot for you. Okay, so that's the most important. I know it is not easy, but just have to move forward. I told you the rigors I passed through to be able to install it on my own system. The work I started around 8 yesterday, and it took me to sleep over it, wake up, sleep until 8 this morning, almost 12 hours. So, but if you are doing the right thing and you are following, honestly, it's not that the journey of 12 hours, you can do it just within 1 hour. Now you have good source of internet, and you are so sure of the stability of the source of your power. So has anybody got to the level of where you will open your anaconda, and there will be a command that will say run as administrator? When you open it, open will be one, run as administrator, is it there? So if you are there, that is take note of the option 2 that is displayed for you. That is, even if your window do not have anaconda already, so, and you want to install it using the prompt, the download http://docconda.io/so, you can type that on your browser. So then you look for anaconda. That is the one I said you should use. So once you are able to open that anaconda, just like what Shalewa did the other time, so you are going to copy copy. It is already displayed for you. Don't forget that I told you that you should open two on the window. You should open two windows, one for ChatGPT that we will be using to monitor what I am doing, what you are watching on YouTube, and there's another one you yourself will be following, will be using to follow up. Who is following up to where you have my own? Okay. If you open too much, then you're supposed to have. You didn't even finish it now. Why is this thing? Already just. That means you have more than one and a half. Yes, because at the end of the day, it's going to slow down your system. You have two storage. So let's see. Now, once you are able to open, get to the, there's one. Where is that? It is already giving options. First option is that if you have amount of that is the story. If you don't have it, if you don't have, follow option whichever one. If you don't have, so this is option two. Just copy this one and paste it in your browser. So if you paste it on your browser, now lead to anaconda. That means it's going to store it. Even if you have it. So now you have installed it, your home. There's another thing. There's another option that this thing provides for you. Now, after the monster, you don't have it at all. So what you will do, or if you have it already, if you don't have it, it's option 1. If you have it, option 2. That means at that level, you will now assume that you already have it. So the next thing is to open. This open is this. Don't forget I said Sorry, what is it? So when you get here, you can see that this one is already installed, and it has told you that it is found in C. That means that is drive C of your system. So now telling you that you should open. There will be one that you should open. So just click on open. It's already open. So come to this your ChatGPT. Copy this code. Come and paste it. That's it. So yeah, click on enter. Enter. Don't type anything. This thing very simple. Enter and wait for it. So anytime this one you can see select. You have to be following up now. Yes, you have to be following up. Anywhere you see, it's already telling you this. Already, you have to be 16 telling you that you have to do this. Do you accept the terms of the channel service is accepted? You have already done that. Now, the next thing for you now is that it's still running. You have to wait. It's going to be the one that is going to direct you. Anywhere you have any green command like this, that means it is successful. If you don't have it, don't be disturbed. Just screenshot where you have it. Paste it back. That this is where you are going to be today. Let's wait for it. Already it is on your system. Once you are able to open, this is what they call the anaconda prompt. That's where you will be pasting that command. You'll be pasting it. So just watch it while it is still change. Yeah, you are also there. Auntie, open your own. Come to this place. Type anaconda. Type anaconda. That will tell you where you are. Click on it. You have not installed it. What happened to has this thing not disconnected? Well, what happened to your connection? You have not installed it. Open this. We're not to now. Open, click on open. Wait for it. Let's see. Just pray it. If it doesn't open, use another. Click on Are you sure? It's asking you, are you sure? Follow it. Next. So, please put this one instead of 25, so that it's not 25. So, come to register. The other one, the other time is not, that was why you were following. So, click on number 2, register. Stop. No solve the environmental spec. Following package will not be installed. The following will not be installed. So, screenshots. Come back to it has opened. Wait, go back to your ChatGPT where you prompted it to store. Store. Try go to that place. Come back. Go back. Go back. Go and paste it. Control V. Enter. Yes. That noise is so what's next? That thing we have it was my own that you are following on YouTube. That means another. Watch. Okay. Accept first of all, accept all. log in first now. You said please. Issue is that Panaro cannot so how to fix this issue? Now that's time to do that. So from here, now man who's taking on this thing is very sensitive. Control. So yes, it's not what it's not going in. Please, if you cannot fix research, and just acknowledge continue. If you have to say yes, wait. If you have to say yes, why? One so because you are asking. Don't I tell you that? Or you listen to go and watch. Just make sure it's on. And see, it's already change will not be effective until the system is completed. You reboot, you refresh. No, sorry, you start. Already whenever you have something like this, and it's already asking you to update and start. So update and run. Start. Are you sure you are following? So I've just displayed up to, but I'm very sure once you are following me up on the YouTube, you should be able to do all these things. Don't forget what I said. Number one, command to install Panaro. The first thing you will do, is that so the first thing you will do is to install. That is option A. Install with Conda. And if you want to install with Conda, this one is telling you that it is the recommended. Please be mindful of not touching the internet, please. Not touching the internet source, please. So install with Conda, which is the recommended, and it is the easiest. So, and that is easy if you have either Anaconda or you have Miniconda on your system. So if you already have it already, just run this code. You open the Anaconda, just go and click on Anaconda in your system, open it, and go and paste this command. Copy this command and go and paste it. That is Conda handles all messy dependency. So by the time you now do that, if you also have option B, the option B is to install with pip. Pip is a Pythonic command. That means whatever, even if you don't have any knowledge about Python, what you should do is that this is recommended. Option B, you should know that option B is the best, because it is going to, especially because you are going to work on bioinformatics. So bioinformatics dependency that people struggle with. So Conda is much easier. So the next one is, where do you get the terminal to type all these commands? Where do you type to type this command? So number one is that you got 4 simple options to choose. Choose which one fits your system. The first option is easiest use of Anaconda prompt. press start or start menu, then you type in anaconda prompt, you open it, then the command you have up here, this command, just copy the first command of option A and go and paste it whenever your terminal must have been open. And the assurance is that that one is going to work perfectly, and you don't need any further configuration. The next thing you should do is just to be following up the instruction or the command it is going to give you in the terminal. So, but if option 2 also now fails, option 1 fails, you now use option 2. And what option 2 is saying is that this you will download, just copy this link and go and paste it on your browser. When you paste it on your browser, then you look for where you have anaconda prompt or where you have miniconda terminal. So you will open it, paste your Panaro install command. So that is a place. So it is this option B command that you are going to paste on when you have opened the second option. Okay, you paste it there, and you click. Each time you copy and you paste, always remember to press your button, which is enter. So after you must have done that, option 3 is that all these after you must have installed your miniconda, there are certain environments that it also requires. Part of the environmental setup that you need to make is that you also need to install some other things that will make your anaconda or panaro, that will make them to be able to function well. And what you do is that when you open your anaconda, instead of you to click on open, go and click. Okay, let me show you this one. So you come here, open the search button. So you now see anaconda, click on anaconda, and when you click anaconda, because I've already set it up, that is why it is opening for me. So instead of you to open, you now open as an administrator. You open as an administrator, you will see it there. When you open as an administrator, you now copy this code. After you must have copied the code, you now click on enter. So after you must have now click on enter, so you allow it to run a bit, then you now restart your system. By the time you are restarting your system, you are going to open what is called Ubuntu. Okay, that is when you type your anaconda already, you are going to see, you can see the icon of Ubuntu. So let's open it. It's already there. So when you open Ubuntu, which is being displayed to you, so come and copy this prompt. Copy this prompt, which is this one. That is number 5. Sudo apt update conda install. Just copy this one. Once you have copied it, you will see this one change to copied. So copy it and come and paste it again in Ubuntu. Okay, paste it again in this. Okay, paste. Paste anyway. So you will see it, then click on enter. Enter. So one funny thing is that you will now be monitoring, because this has been installed already. That's why this one is being displayed. Okay, channel terms for this. Okay, so don't forget we are following up with what I have on ChatGPT after number 5 now, which is going to allow you this. So you go to 4. So if you cannot go to option 4, that is you will now type control. You type control, just copy this. Okay, control alt 3, control alt 3, alt T, Control Alt T. So it's going to open it for you. Then you copy this code, this same code, conda install C and so. So come and paste it here again in your terminal. Paste it. Control V. You can see, then enter. You will see how it is going to react. You can see the current user does not have the right